Genetic engineering can involve electrophoresis, polymerase chain reactions, bacterial transformations, and DNA sequencing. So we start with structural genomics here. And in 1990, the Human Genome Project began. And in trying to figure out the sequence of the human genome, other organisms their genomes have been discovered, like the first bacterial genome, and yeast, and mice, and E. coli, and planaria, and mustard plants, and fruit flies. And eventually, 13 years later, we end up with this finished version of the human genome. And it's from this structural genomics where we find out the genes and the sequences of all these other organisms that allows us to also get into functional genomics and how genes work and what specific genes make specific proteins and what they do. So we can study mice genomes to understand the development, treatment of human genetic disorders, planaria, regeneration, neurological diseases, fruit flies, a lot of development, neurobiology, mustard plants, getting into the flowering plants, algae with photosynthesis, response to stimuli, roundworms, getting into apoptosis, aging and disease like cancer and diabetes, yeast, we start getting into just the proteins of eukaryotes. But ultimately, we can understand functional genomics through the creation of knockouts. So it's evolved to start using CRISPR-Cas9. So we have these CRISPR sequences that can create target genes, and that or RNA sequences to find target genes. So we have this Cas9 protein enzyme that will cleave DNA. So when you give a sequence here, a CRISPR sequence, to a specific target gene that you want to study, the enzyme will cut apart the DNA that you're targeting, and that can create a small deletion, or you can insert a point mutation of donor DNA, and ultimately whatever you are targeting is now basically knocked out or turned off. So what happens is they go through that, they create a knockout genetically engineered sequence of DNA, and you can take embryonic stem cells from the model organisms you want to study. You add in that, you genetically engineer that knockout, you disable a gene, and then you take this embryonic stem cell that's been engineered, put it back into an embryo, and put that into the uterus of a host female, and the mice that are born afterwards are going to have that gene disabled. So in that process, you can study treatment. So if you basically give mice cystic fibrosis, you can then understand the development, but then try different therapies on the mice to see if it alleviates any of the symptoms. You can also take disease model organisms and knock out specific genes to see if that affects the toxicity of that disease, and ultimately that gives you a target from which to develop drugs. So as we keep going, we can use these sequences to do comparative genomics, not just functional genomics, to see the relationship between species. So in this case, it's looking at human hemoglobin and comparing it to other vertebrates to see how many differences are. So the greater number of differences, the more distantly related the organisms through phylogeny. So when we take a look at this, we can hypothesize about the relationship between species because of being warm-blooded, the number of chambers in their heart, and we can have different hypotheses about these different species and which one is most closely related. And then we can actually compare DNA sequences. So in this sequence, we're looking at the mammal and the bird, looking at the differences between them. And this one is the bird and the lizard. Between the mammal and the bird, there's 13 differences. Bird and lizard, there's 7 differences. So that leads us to believe that the bird is more closely related to the lizard. Another idea behind genetic engineering is the concept of PCR, or polymerase chain reactions. So we have this thermal cycler, and you have to load different things in there. Free nucleotides, primers, buffers, polymerase, and ultimately your DNA sample. So what's going to happen here in this polymerase chain reaction is it's going to amplify whatever sample you put in there. So initially, typically if you would crank the temperature up, you would not only split apart the DNA, but you destroy the enzymes that are going to do the copying. So this Taq polymerase here is actually from heat-resistant bacteria, so then it can still do the replication that's necessary without being destroyed by this high temperature. So you crank the heat up, the DNA splits apart, ultimately it's then lowered way down, and these primers are able to grab on, and then you put it at the ideal temperature for Taq polymerase to extend those primers and grab on, and it's making taking those free nucleotides and adding on to the new DNA sequence. And if you just keep running this over and over and over, every time you run it through a cycle, it's going to double the amount of DNA available. So this is a very simple process to create and amplify a tremendous amount of DNA. Now, how that might be used, you can actually take DNA from the environment. So you can take a sample of sediment, a sample of water, even a sample of feces, and determine what organisms may be in there microscopically, or what DNA has been shed into that environment. So when things die, their DNA is still present in the environment, and even our own presence, we lose epithelial cells, and that's going to contain DNA that would be in the environment. So again, it takes these, it extracts the DNA from the sample, and then it goes through the PCR and makes a tremendous amount of copies, and then it can be sequenced, and then you can figure out what's actually there. So from that, you can actually determine the organisms in an environment simply by analyzing the DNA shed in a water sample. As we keep going, we have this idea of restriction enzymes. There's going to be these specific sequences that are sought out by these enzymes, and it's going to split apart at that sequence. So that gets into electrophoresis. So if you have a sample of DNA and you have a restriction enzyme that's looking for this specific sequence to cut, you would put the sample DNA in there, and whenever it came across that particular sequence, that would get cut. And you'd get these different sized segments of DNA. Now, from different people or different organisms, those repetitive sequences are going to show up at different locations. So not every single DNA sample will get cut at the exact same spot, because in this DNA sample, the sequence is here, 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 and here, but in this one, it's at different locations. So you take all these different samples and you chop them up at specific sequences, and you end up with different bits of different lengths. So ultimately, when we talk about gel electrophoresis, we're taking this agar, we're pouring it into a cast with a comb. It's a liquid and then it solidifies, but when you pull this comb out, there's these little wells. And it's in those little wells that the samples can be injected into. So you take that sample and you inject it into that well. You take that sample, inject it into the next well. You take the next sample, put it into the next well, next one into the next well, and then ultimately it's run through a chamber that is hooked onto an electrode. This electrode, because these different segments of DNA have different charges and they're different sizes, they're going to get drawn through the gel. And little segments will get pulled much faster, and the big long segments will get pulled much slower. Ultimately, you end up with this gel that you can analyze. So if we look at this, you took some crime scene evidence, some DNR, and you do your PCR and you cut it up, and you end up with 2 bits of DNA. that get pulled into these locations. Well, if you have two different suspects, you can take their DNA, chop them up through the same restriction enzymes, and then run them through the gel, and you can see which DNA matches with which suspect. Now again, when we look at a crime scene, it may be unknown DNA, so you would compare this DNA from the crime scene to the victim and to the suspects. And as you can see here, it's not, this DNA from this crime scene does not belong to the victim. It does not belong to suspect 2, but it matches with suspect 1. Another way that gel electrophoresis can be used is through parental, figuring out parents. So if you have an infant with an unknown father, you can compare that infant to the known mother. And when you look at this, the infant is going to be sharing DNA from both parents. So what we're looking for is not the DNA sequence that the infant has with mom, because this sequence from the infant came from mom, this came from mom, this came from mom, this came from mom. We want to see the other parts that didn't come from mom and which male it might be associated with. So we can see this spot right here. Male number 1 does not have it, and neither does female. So male number 1 cannot be the father. However, male number 2 does have that and could have contributed that section of DNA to the child. Again, with this, it doesn't match up with mom, but it matches up with male 2. Doesn't match up with mom, matches up with male 2. So again, getting back to this idea of restriction enzymes, really important to be able to cut apart things at specific locations. So now we're getting into bacterial transformation. So we can take a plasmid from a bacteria, cut it with restriction enzymes, and then we have ligase which ties in the DNA, and we can cut and take out an insulin gene from a human, put it into bacterial DNA, have that recombine and make recombinant DNA, and then it can get sucked up through transformation back into the bacteria, and then we can make copies and have gene cloning. By having this go through division, you can ultimately end up with clones of that particular gene, and then the bacteria would actually go through the process of transcription and translation to make the human product, like insulin. So when we're talking about bacterial transformation, again, bacteria can pick up plasmids or naked DNA from the environment. So we can ultimately genetically engineer a plasmid, and then we create an instance where the bacteria might suck up DNA from the environment, which can involve heat shocking. Get it really cold, get it super hot, get it cold again, and it's that transfer of temperature that can cause it to suck up DNA from the environment. Now again, this isn't perfect, and not every single DNA or not every single bacteria is going to pick up every single plasmid. It's not 100% successful. So what we want to do is just focus on the actual recombinant strands. We only want to focus on the bacteria that have picked up the recombinant plasmid. So we end up putting it onto agar plates, and a lot of times, to make sure we know which ones have it, if we just grew all the bacteria on regular agar, they would all survive whether they have the plasmid or not. So sometimes the plasmid that we add in doesn't just have what we want, like an insulin gene, but it'll also put in there resistance to an antibiotic like ampicillin. So these genetically engineered bacteria, if you put them on an agar plate that they would usually grow on, but you also put antibiotic on here, everything that doesn't have the plasmid is going to die, and the only ones that survive are going to be the transformed bacteria that have the recombinant plasmid. So this idea it brings us to GMOs, so genetically modified organisms. We think about this a lot of times with corn, and the corn has been genetically modified so you can spray Roundup herbicide on it and not kill the corn, but kill everything else. So again, when we come back to this pathway, when we're talking about tryptophan and gene expression, the operon, it would take these precursor molecules and ultimately create the enzymes necessary to transform the initial shikimic acid into the amino acids. Again, plants aren't eating proteins and getting amino acids that way. They have to create their own amino acids from scratch. So what Roundup does is it's made of this glyphosate, and what it does is it inhibits this enzyme in plants. So if you spray Roundup on any plant, it's no longer going to be able to make these amino acids. And if it can't make these amino acids, it's going to die because it can't make the essential proteins necessary for life. So genetically modified corn, what it has done is it uses a bacterial version of this enzyme that glyphosate will not inhibit. So ultimately, you can spray Roundup on genetically modified corn because it has a special enzyme that won't be broken down, and every other plant that gets in contact with it that has this enzyme will die. So how are they actually genetically modified? So we take a bacteria, we take out the plasmid, we genetically engineer that new version of the gene for the enzyme that won't be inhibited by glyphosate, and then we transform that bacteria and get the bacteria to suck it back up. Now, this bacteria that we're dealing with here is an agrobacter. And it kind of goes through conjugation, but instead of conjugation with other bacteria, it kind of creates conjugation with plants. And it can take plasmids and inject that plasmid into the plant, which will ultimately become part of the host DNA. So then we can regrow that plant, and now all of these plants have that genetically modified plasmid and embedded into their host DNA, which will make them able to have the enzyme that can't be broken down by glyphosate. So we take those plants, we grow them in a culture, and now we have genetically modified plants. Last example here, we can also do that with animals. So we end up with transgenic animals. We can take a plasmid, genetically modify it, so it has a growth hormone gene from this Chinook. But then the promoter, it can take from this other ocean poutfish, and ultimately this promoter is super active. So then if you put that promoter in front of this growth hormone, it's like it's going to always have that gas pedal stuck down in order to keep on producing growth hormone. So we genetically engineer this plasmid. They transform it into bacteria. Billions of copies are made through gene cloning. And then ultimately we can snip out those segments again, mix them with fertilized fish eggs, and ultimately you end up with this transgenic salmon. And this transgenic salmon can grow 3 times as fast as a wild salmon and with less food. So this process is that, again, to try and control this process, all of these transgenic salmon are female and sterile. So the only way you can create these is through this process. So again, we have genetic engineering like electrophoresis, polymerase chain reaction, bacterial transformation, and DNA sequencing that all lead to the uses of biotechnology.